Today we're going to talk about the mechanisms of tolerance that we discussed last time. But specifically, for the most part, we're going to talk about some of the ways that we understand those mechanisms of tolerance can be broken to lead to autoimmunity. And so we're going to talk about a few different scenarios that have been shown to lead to autoimmunity, as well as some other details regarding autoimmunity. Because I know you guys and know the way classes tend to go, I'm sure that there will be absolutely no questions about any of this, and that we'll just, speed right through it all. But hopefully, I have a feeling you might find some of this rather interesting, so that's what I wanted to point out. So if you remember last time, we talked about 6 mechanisms of tolerance that we think about in terms of peripheral tolerance. So we had, this is terrible shot. So we had central tolerance, versus peripheral tolerance. And within peripheral tolerance, we had clonal deletion. We had clonal energy. We had clonal suppression. We had clonal hopelessness. Clonal and we had clonal ignorance. Which is also sometimes known as immune privilege. So these were some of those mechanisms of tolerance we talked about last time. And I left off talking a little bit about clonal ignorance or immune privilege. If you recall sort of the classical definition of clonal ignorance or immune privilege is that there are certain anatomical sites, the brain, the eye, the testis, and the uterus being the most famous of them, where there's sort of like an anatomical barrier to exclude the immune system because the price of immunopathology, the price of an immune response going wrong there was just too high. And I even showed you some data about the fact that sometimes we can have infectious pathology in those sites that may look different than the rest of the body, perhaps because of this sort of lack of immune response. But what I also told you was that our understanding of immune privilege or clonal ignorance has definitely changed a bit over time. We used to just say it was the immune system in these areas just doesn't see any stuff. And we now know that that's probably not the case in quite the same way. And one of the examples of how we really started to modify our understanding of immune privilege had to do with multiple sclerosis, specifically with actually a treatment for multiple sclerosis that I'll tell you about on the next slide. So there's lots that can be said about multiple sclerosis, but just to give you a few pieces of information that are sort of relevant to this this situation. Here at the bottom, you can see a healthy nerve cell, and you can notice that we've got this sort of, the nerve cell is in orange, and there's this blue stuff around the outside of the nerve cell, particularly going down the axon. That blue stuff is myelin. That's known as the myelin sheath. And basically, the way you can think about the myelin sheath is that nerve cells work by electrical conduction. So there's actually electricity going down this axon to go from one cell to another. And we want to make sure the electric charge goes in this direction and doesn't go like out in this direction to the side. We want it to go straight down the axon. And so we put a coating that the electrical charge can't go through around the outside of the axon, so the charge can only go straight through the axon. It's honestly the exact same reason why this wire has plastic around it. The idea is that we don't want the charge to go sideways and come out of the wire and shock me when I grab it. We want it to go straight around the wire. In the case of multiple sclerosis, there is destruction of that myelin sheath. There are holes in the plastic around the wire. It's not plastic, it's actually lipids, but you know, whatever. And so you can see we've got this demyelination. As a result, it's the same thing that we would really see if we have a wire that has the plastic kind of coming off in different places, which is that we start to see random shorts in the electrical connection. Every so often the electricity starts going sideways and we start, getting shocks, and we don't get good transmission. And so we have some neurologic disease because we're not able to get good electrical transmission straight down this axon. And we know that the reason why we see this demyelination, why we see these breaks in the myelin sheath, is due to T cells going into the brain and actually killing the cells that add the myelin or otherwise destroying the myelin sheath. T cells are often thought about as a main contributor to this loss of myelination, though we can imagine antibodies and some other molecules being a part of this as well. And so some immunologists took this information and they came up with a hypothesis for a way to cure multiple sclerosis. They said, well, it looks to us like the problem with multiple sclerosis is that we've got these T cells in the brain killing the parts of the brain that are involved in the myelination process, and thus destroying the myelin sheath. And normally the brain is immune privileged, so the problem must be that T cells can get into the brain where they usually can't. So they said, our hypothesis is, let's make a drug that blocks T cell trafficking so that T cells can't go to the brain. So they specifically came up with an antibody, which you can see here in blue, that bound to an integrin. So you've got this blue antibody binding to the alpha-4 beta-1 integrin, which is the integrin that T cells use to go into the brain. And so they said, if we just block T cells from going into the brain, they can never go make damage to the myelin sheath, and we all live happily ever after, and people don't have multiple sclerosis anymore. The trade name of this antibody as a drug on the market is Tisabri. And when Tisabri was used to treat multiple sclerosis patients, it was pretty revolutionary in terms of how it worked for those patients. So if you looked at those patients, in the blue line you see the placebo patients, and so what you can notice is that over time their brain gets an increasing number of multiple sclerosis lesions, like they're getting increasing amounts of damage. When we treat these patients with this integrin, so T cells don't get into the brain anymore, we don't see those lesions. So we basically stopped the multiple sclerosis, like life-changing for these patients. Absolutely life-changing for these patients. Huge deal for these patients with Tisabri. And so that was great, except then some of the Tisabri patients started to die. And we realized that there was a problem with this whole idea. Because in fact, when we looked at these Tisabri patients, we found, and so here you can actually see Tisabri was FDA approved in 2004, and it was pulled off the market in 2005 because of these disease cases and these patients dying. And these patients, as you can see from this timeline, died of something called PML. And PML, you don't have to get stressed about what PML is or anything, but PML is officially progressive multifocal leukoencephalopathy. It is a disease that is basically a neurodegenerative disease. In some ways, its symptoms looked really similar to multiple sclerosis. And when people sort of investigated what was going on, they found that, some of this was known before, but all the pieces got kind of put together. PML is caused by a virus. Virus is known as JC virus, which is actually named after the first patient who ever had it. I believe his name was John Cunningham, but I don't remember, but I know it's JC. JC virus. And what people realized is that JC virus is actually in the brain in most healthy people. You guys may have JC virus in your brain right now. And that virus is actually being kept under control by your immune system. So it's not damaging your brain. It travels pretty easily through blood. It's one of the viruses that's found in a massive percent of people.
Like you just picked it up and it's just there?
Yeah. Like I said, it's one of the few viruses that just pretty much that a large portion of people have. And usually JC virus is just reproducing in your brain, causing no issues. But it turns out, if you stop T cells from going to the brain, now JC virus can reproduce out of control because there are no T cells going to the brain to stop it. And so we have these patients whose multiple sclerosis was stopped because we stopped T cells going to the brain. But as a result, viruses that were already in the brain kind of went out of control. And these people died of PML. And so then they started doing things like, let's basically, before someone gets Tisabri, we'll check and see if they have this virus. And we'll only give it to people who don't already have this virus in their brain. It's been a whole thing. It's been on and off the market a bunch of times. But the point, or the reason why I want you to know about this is that this is one of many examples where we said, I think immune privilege might not work the way we originally thought. Because the idea was the brain had no T cells, and that was what immune privilege meant. And here we blocked the pathologic T cells in the brain. We blocked T cells going to the brain, and we learned, those T cells were actually doing a job. We actually needed those T cells in the brain. They were really there all along. It wasn't like they were totally excluded. So while the brain is an immune privileged site, it isn't like it had absolutely no T cells. Because if we do something that makes that brain have absolutely no T cells, bad news happens. Instead, the way that we really think about immune privilege now is that these sites have kind of modified immune systems to reduce the amount of immune responses, or in some cases, to push immune responses more towards like a Treg phenotype. So we don't see as much inflammation and we tend to see more Tregs. When I was at that conference I went to in October, there was one talk I went to where someone was talking about special Tregs of the eye. And so the idea might be not that the immune system is completely excluded from these locations, but that it is different in these locations to try to eliminate disease. And one thing that I will mention about that is that again gets to this question of how much of this is really Tregness, how much of this is sort of related to clonal suppression? If you remember, and you will see so much more of this today, which is kind of why I'm mentioning it, when I talked about clonal deviation a little bit before, clonal deviation was originally talked about as balance between Th1 and Th2. And we now have realized that sometimes it's like balance between, say, Th1 and Treg or something like that. And so that's another one where, like, maybe it's kind of leads into clonal suppression a bit. And so how much of this is related to things like Tregs and clonal suppression, and how much overlap there is an interesting problem. So I've already shown you this slide once. I will probably, depending on timing, show it to you one more time. So again, you're not expected to get every little piece of information here at all. When I showed you this slide before, I showed you this list of common autoimmune diseases and their prevalence. And what we thought about at that point was that we thought about central tolerance. And we thought about the fact that central tolerance had a bunch of sort of cracks. There were a bunch of places where we could imagine cells kind of going through the cracks of central tolerance. And we looked at these numbers in terms of prevalence of these autoimmune diseases, and we said, whoa, those prevalences are way more rare than we might have expected, given that everybody has these flaws in central tolerance. And of course our answer was, and that's why we have peripheral tolerance. Now we've talked about the mechanisms of peripheral tolerance, and we can look back at these prevalence numbers. And now what question might you have about these prevalence numbers? Yeah, David.
Why are they so high if there's all these?
And now it's like, well, wait a minute. We have 6 overlapping things protecting us. How the heck does it go wrong this often? Now you might look at this and say, I think these are too common. Whereas before you thought they were too rare. And so what I'm going to move into now are kind of thinking about ways that tolerance can be broken. And so some of the ways that these mechanisms of tolerance that we've been talking about can go wrong. I have 2 examples that I want to tell you about where the problem is a problem with central tolerance. So there are 2 famous diseases where the problem is central tolerance. And one of the things that's important to know about these 2 examples that I'm going to give you of central tolerance breaks is that in both cases, the patients have, as it says at the top here, systemic autoimmunity. What that means is that these patients don't have like one autoimmune disease, like one of the things on this table. These patients tend to have multiple autoimmune diseases together. They have many different organs, many different self-antigens that are being attacked here. And if you think about it, you can imagine that if something's going wrong with central tolerance, it's going to be a lot of cells that are going to be slipping through the cracks. And so you're going to tend to get many cells. In both cases, I'm telling you a T cell example. You're going to get many T cells that are going to leave the thymus inappropriately. And so you're probably going to develop responses against many antigens. So one of these diseases is a disease called APSED. The abbreviation is up there at the top. I've already actually told you about this earlier this semester. APSED stands for autoimmune polyendocrineopathy and candidiasis with ectodermal dystrophy. I'm going to say it. This disease is a result of patients having a mutation in AIR. So those patients are not, don't have the AIR protein in their thymus, so they're not making all of the self-proteins that they're supposed to make in the thymus. And so they cannot test T cells against all of the self proteins that need to be tested. So we're going to not negatively select some cells that should have been negatively selected, and those cells can go into the periphery and lead to autoimmunity. And what you'll notice is that if we look at these patients with APSED, they have kind of everyone has sort of a different combo of autoimmune symptoms. So this table on the left is showing you the percent of patients with each of these different symptoms. So you can see some of them will have thyroid issues with hypoparathyroidism, some will have adrenal failure, some will have ovarian failure, some will have diabetes. A lot of them have this weird phenotype of their fingernails, which is shown here. That's the nail dystrophy. Pretty much all of them have recurrent yeast infections, and so there's the candidiasis. But there are a lot of different things going on, it seems, in these patients. And the key to note is this is an issue with central tolerance. The other kind of famous example of a defect in central tolerance is a disease known as IPEX. And you can see IPEX up here. To spell it. IPEX stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. So again, we've got a whole bunch of things. Immune dysregulation, polyendocrinopathy. It seems like in both of these cases, the endocrine system gets really messed up. Like the autoimmune disease really destroys the endocrine system. And you can see that this is X-linked, so this one is mostly found in boys, because the gene that is a part of this is on the X chromosome. So you really are only going to be in a situation where you have two bad copies or one bad copy if you're male. It's unlikely that you're going to get two bad copies if you're female. The gene that is the problem here, the gene that is mutated, is FOXP3. And so FOXP3 is on the X chromosome. There are some boys who will not have FOXP3, and so they cannot make any Tregs. And so they really never develop Tregs. It's a pretty big problem with central tolerance. And what you can notice in this, I pasted at the bottom, the disorder manifests with autoimmune enteropathy, so autoimmune GI symptoms. There's destruction in the GI tract, eczema or other dermatitis, again, nail dystrophy, autoimmune endocrinopathies, so messed up endocrine system, autoimmune skin conditions like alopecia or pemphigoid, massive enlargement of the secondary lymphoid organs, diabetes, food allergies, and infections, and most children with this die within the first 2 years of life, unless they get a bone marrow transplant of bone marrow with FOXP3. And so what I, again, want you to notice here is this one is usually a defect in central tolerance. You can't even make those Tregs in the first place. And so you're going to see issues with T cells of many specificities, not just one. And you're going to see this sort of diseases across the body, not just one kind of autoimmune disease. So we can then switch to thinking about peripheral tolerance and defects in peripheral tolerance. A couple of sort of big picture things to note about peripheral tolerance breaks and the way that they tend to lead to autoimmunity is very often these peripheral tolerance breaks will happen to like a particular T cell or a particular B cell. And so you may not have, T cells responding to tons of antigens that are the problem. You may have kind of the one T cell that slipped through the cracks. And as you will notice in some of these tolerance break examples, in some ways it's like a T cell that slips through the cracks because of like when it came out of the thymus relative to like when certain other events happened. A lot of times people will, be like, well, why do I have this autoimmune disease? I actually have a friend with a child with autoimmunity, and she like asked me, and she's like, what did I do wrong? And honestly, as I hope you will see, but it also just makes life as an immunologist sort of hard, in a lot of these cases for the tolerance break, there is a definite role for bad luck. Of like, if this bad luck thing happened to allow this cell to slip through the cracks. And usually we're gonna see sort of one organ, one disease in these peripheral tolerance breaks. We'll talk later about some of the roles for genetics in autoimmunity, but I will also say that very often we see a role for infection in autoimmunity. And there are a lot of ways that we can think of infections potentially leading to autoimmunity. One of the things that you can notice here is that there is sort of an HLA association. So some of this has to do with you had to be unlucky enough to get the right infection and to have, or to get the wrong infection, I guess, and to have the wrong type of MHC and to have to be on the wrong day. And so you can see there's a lot of this role for bad luck here. Again, we can talk more about the MHC type association later. But you can see there are a lot of associations between infection and autoimmunity. One reason I tell you this is I think that every one of my examples kind of involves an infection happening. But you can also see that my title here has a question mark at the end. All of the examples I'm going to show you about are examples that involve infection. But this association can be extremely hard to prove because, of course, you need to show that people who got Coxsackie-A virus, it's not everyone who gets Coxsackie-A virus who gets type 1 diabetes. It's some people who got Coxsackie-A who also happen to have this HLA association. And even then, it's not 100%. And so that becomes kind of difficult. But the other place where this becomes really, really difficult is that usually it's not like you get Coxsackie-A and you have a particular HLA association on Tuesday, and you end up at the doctor's office with type 1 diabetes on Wednesday. That's not how this looks. Generally, the way that this looks is that you get some infection at some point in your life. Maybe you get this infection when you are 10. You get this infection when you're 10. And as a result of that infection, one of the events that I'll show you on some of the next slides happen, and you get a T cell that gets messed up for peripheral tolerance. So here's going to be a messed up T cell at age 10. And that messed up T cell reacts with beta cells in your pancreas and kills the beta cells in your pancreas, which are the ones who make insulin. It might take 5 years for the T cells to damage enough beta cells in your pancreas for you to feel sick. It might not be one beta cell gets destroyed and you feel different. You might need to have many beta cells destroyed before you actually feel this. There's probably, in a lot of cases, a fair amount of the autoimmune damage happening before you ever show up at the doctor's office. And because of that, the doctor has to figure out, well, it would be like, what MHC type is it? But also, do you remember, because Coxsackie tends to be a respiratory virus, did you ever have random sniffles 5 years ago? And did that lead to your diabetes? And so those relationships are really hard to actually make. There are some ways that we can make these associations, but I think that most of the time the infection that might lead to these tolerance breaks will be an infection that we're never going to be able to actually find and tie back. Again, it's a matter of unluckiness. There is a really famous paper, and I meant to look at the paper because I haven't read it in a while before this, and I totally forgot. But there's this one infection, multiple sclerosis association that people have sort of hypothesized about for a long time, and there's a paper that actually did a really good job epidemiologically to show it. And if I remember correctly, and I will look back at the paper to get this number right, I believe they had to look at 8 million people before they could get the statistical power to actually show the association. And so these associations are really, really hard to show. And so again, remember that many of the events I'm going to be showing you here are sort of the initiating event. They're the event that makes the first cell not have peripheral tolerance. But that doesn't mean that you're going to know it that day. It's just going to sort of start the process of this response, and then other aspects of sort of physiology are going to play a role before you actually show up at your doctor's office. One thing that we know about that can be important with some aspects of tolerance breaks is a phenomenon known as epitope spreading, this is sometimes discussed as autoimmune amplification. And here you might see we have some infectious agent that looks like a red starry virusy thing. And we're going to make a response to that, as we should. And then that T cell, once that T cell's been activated, is going to go out and find cells that have the pathogen. This is all the way it's supposed to work, right? This T cell is going to go out and kill these infected cells. Again, this is the way it's supposed to work. But when it does that, some self-proteins that were inside those cells are going to get released when these cells die. And it's possible that those could be picked up by phagocytosis, by a dendritic cell, for example, perhaps presented on class 2, and then they could start to act as antigens. And so maybe when they were sitting happily ever after inside a cell, they weren't really getting phagocytosed, they weren't really getting presented, we weren't really seeing a lot of responses, but maybe in this situation when they get released, and they're getting released as they should when the infected cell gets killed, so it's sort of just bad luck that those cells happen to have an infection, then maybe we start to see this sort of amplification of these epitopes.
Just to clarify,
Yep.
the peptides being released, are they the ones that were from the infection, or just in general peptides?
No, it's just other self-proteins that were being released. And some of that could even relate to some of the other mechanisms that I want to show you. I'm not going to say a ton about ways that you break clonal deletion or ways that you break clonal helplessness. In both the cases of clonal deletion and clonal helplessness, part of that mechanism was that a cell dies, right? We either deleted the cell or the T cell got deleted so the B cell doesn't get help. So if you really wanted to break one of those two, you'd have to do cell reincarnation. You'd have to take a dead cell and make it alive again. And that's not really a thing. But some of the other ones, we have some pretty obvious mechanisms of how we can break. And so one of the really important ones is clonal anergy. And I will say a couple of you guys were kind of making some faces with the epitope spreading and perhaps had some questions about the epitope spreading example. I could, if I were going to sort of make some arguments, I could say that if I took one of the anergy mechanisms and one of the deviation mechanisms and added them to that epitope spreading, it to me would be more clear. But I haven't told you those two things yet, so I can't put them together yet. But if that's something that you want to talk about later on, we can do that. So here is the information about clonal anergy that we talked about before. So remember that we have this situation where our antigen presenting cell is presenting antigen on MHC. So we've got this MHC plus peptide, and that antigen presenting cell has also seen a microbe using a PRR, and as a result has made B7. And so we're going to turn that T cell on, giving a signal 1 and signal 2. If we're just presenting antigen without the B7 because there was no infection, there was no PRR stim to tell that antigen presenting cell to make B7, this is likely a case of self-antigen turning on the T cell, and that T cell becomes anergic and stays off later on. So there is a little bit of a problem with this scheme. And the problem has to do with the fact that T cell receptor and CD28 and B7 and MHC class 2 are all integral membrane proteins. They're all transmembrane proteins. And the way that integral membrane or transmembrane proteins work is that they diffuse throughout this plasma membrane. What that means is that MHC class 2 and B7 are not always necessarily next to each other. And the thing that's important about this, and you'll see this on the next slide, is that this cell, this antigen presenting cell, probably, in fact not probably, definitely does not just have one MHC molecule on its surface with one peptide. It's got a bunch of MHCs on its surface, and each one has a different peptide. Let's imagine for a second, I'm going to make the numbers easy. Let's imagine for a second that there are 100 MHC molecules on the surface of this cell. And this cell is infected with some kind of microbe. If you had to guess, and yes, we are 100% guessing and making up a number here, how many of those MHC molecules that are on the surface of the cell are going to be presenting peptides from the microbe? Okay, so Shelby thinks 1. Okay. Is the answer 100, do you think? Is it all of them? No. There's going to be something else presented. What's the something else? What is it, Brenna?
So?
It's going to be some kind of self-antigen. So let's imagine sort of a scenario. Let's imagine that there's a situation where 99 of the 100 are presenting pathogen, and 1 of the 100 is presenting self. This B7 doesn't know it's not supposed to go next to the MHC that's presenting self. It just goes next to an MHC. And so suddenly, where we shouldn't have been turning on a T cell, because we shouldn't have had B7, this antigen presenting cell did in fact see a pathogen. That pathogen is not related to the MHC it might be presenting, but it did see a pathogen, so now it's making B7, and it is accidentally activating self-antigen. And so you can see that here. So what we should have is we should have this situation where, at rest, we're presenting just self-antigens. They interact with T cells, and that makes the T cell anergic. It gives that T cell self-tolerance. But perhaps we alternatively have a microbe that activates that antigen-presenting cell. That antigen-presenting cell will present some self-antigen and some microbe antigen, some percentage of each. You can argue about what percentage. I don't care. The answer is that it's some percentage of each. And now, because this antigen-presenting cell has seen a microbe, it starts making B7. That B7 can be next to the MHC presenting the microbial peptide to turn on the T cell we want to turn on. But that B7 could also get next to the self-antigen that's being presented and turn on that self-reactive T cell. And so in some ways, what you can think of, because then I know sometimes people look at this and they're like, we should all have autoimmunity all the time because of this. But realistically, this T cell who's getting activated here has to be a naive T cell that just came out of the thymus that has not yet been energized. Most of the time, the first time that T cell sees its antigen, it's going to get energized. It's going to go through this self-tolerance. But if it just so happens, on the wrong day, you make a new T cell from your thymus, and that T cell happens to leave the body, the thymus, and happens to go to a location where its antigen is getting presented by a cell that also got infected by something else. If those unlucky things happen, then you're going to turn on a T cell, and that's self-reactive. You're going to then have a T cell that can destroy and lead to autoimmunity. And so this is one big way where we could break clonal energy. Yeah, Brenna.
I just want to make sure. So the epitope searching refers to helplessness and deletion, but this is its own.
Epitope spreading can actually refer to a few different things.
Mm-hmm.
Helplessness and deletion really don't have ways to break them.
Oh.
So epitope spreading I didn't put with a specific thing. I did epitope spreading and said it's a thing that can happen. It could actually be related to a bunch of this other stuff. And then I said helplessness and deletion don't really have ways to be broken. So this is one way that we can have energy get broken. There is a second way that we can also have energy broken. And this one actually has like a fun name. The one I just showed you a second ago, everyone always sometimes on exams, students try to write a fun name for it. I don't know, inappropriate B7 sharing or something. There isn't a perfect name for it. This one has a name. And the name for this one is called molecular mimicry. And so what you want to, again, think about is MHC peptide interaction. So here you can see two MHC molecules. You can see that perhaps they have differences in their peptide binding cleft. So this one binds to reddish peptide, this one binds to white peptide. But they're binding to only part of the peptide. Part of the peptide is facing up towards the T cell. And maybe the part of the the part up here at the top from the pathogen and the part that is a self-peptide look the same. Maybe sometimes we have self-peptides and pathogen peptides that are really similar. If that happens, the same T cell might see both of these MHC plus peptide pairs. As far as the T cell doesn't see this bottom part down here, which is actually the only place we have things be different. The T cell only sees the top. And so maybe we turn on a T cell correctly in response to this pathogen peptide. We want to get rid of the pathogen. Now that T cell is on, and it's on all the time, and it doesn't need signal 2 anymore. And so maybe at the beginning, we recognize the pathogen peptide, we get rid of that, we're happily ever after. That T cell now is an effector T cell. That T cell is on all the time. And so that T cell might also bind to this self-antigen that looks really similar. It is in fact a molecular mimic of the pathogen peptide. And that T cell may now kill this self cell. And so because of the sort of biochemical similarity, perhaps in this case between the pathogen peptide and the self peptide, we're going to kind of see this potential issue happening. Yep, Jonah.
Is this what's going on with the goal of infection with autoimmunity when it comes to a virus, or is that?
So this could be, but you could also imagine there's a pathogen here.
Yeah.
And so a lot of these examples, there was a pathogen here too. So a lot of these examples could involve some kind of infection. So what I wanted to point out was that we can't very often actually prove that there was an infection, but if you look at most of the mechanisms that we think about, we usually have an infection involved. But if somebody has an autoimmune disease, we're probably not ever going to be able to say it was such and such infection that got you many years ago. Maybe we could say, hey, if we vaccinate people against more infections and we stop them from getting those infections, then we're going to prevent autoimmune diseases. But we kind of have to have some of this, now that we know about particular viruses that might be involved with multiple sclerosis because we looked at 8 million people. Now maybe we have evidence to say we should vaccinate against this to prevent multiple sclerosis, but that's still a whole other can of worms. But so the idea that I wanted you to see is that there is often an infection, and I think every example I have is going to have an infection involved, even if we can't always like pinpoint what infection happened to a person. And so similarly, this is really all about this sort of phenomenon that's known as cross-reactivity. And so the idea is that we might have some pathogen peptide and some self-peptide that look really similar. Maybe they have, 7 of the same 8 amino acids, or they're really, really close together, such that the same T cell that recognizes one is going to recognize the other. And so I mentioned sort of the diabetes example. One of the things that we think happens is that in childhood, someone might have a particular viral infection in the upper respiratory tract. We turn on T cells, B cells, we do all the things we need to do to get rid of that respiratory virus. The kid grows up, feels better from the cold. But one of those T cells also might recognize pancreatic beta cells. And so now that those T cells got turned on in response to the virus, they're effector cells. They don't need signal 2 anymore. They kind of don't have the same kind of checks and balances on them that they used to. They go into the pancreas, they realize, oh my gosh, I recognize pancreatic beta cells, and they kill those beta cells to the point where the child will eventually lose all of their beta cells. This may take a certain amount of time. And in fact, the situation where T cells have killed all the pancreatic beta cells means you don't get those cells product anymore, which is insulin. This is type 1 diabetes, and sort of how we think that works. We also have realized that in some cases, particularly with these molecular mimic situations, sometimes there'll be a situation where the T cell will actually bind to the MHC plus peptide just at the wrong angle. And suddenly, a self-peptide will look like a foreign peptide because the binding angle between the T cell and the MHC is wrong. And so this is actually showing you a situation of what it looks like. You've got in blue the MHC, the green is the peptide, and the red and the yellow are the TCR alpha and beta chains. This is sort of how the binding should look when a T cell is responding to influenza. But if we actually look at what happens in some MS patients, you can see that the T cells are binding to the MHC plus peptide just at the wrong angle. And so what maybe in the thymus was sort of one kind of interaction, now it looks like a slightly different interaction because of that difference in binding. Why the T cell might do that, we don't really know, but for those of you who kind of think about the biochemistry, this is a simple way that self may look like foreign, or that foreign might look like self. And those examples that I just showed you are examples that were with T cells, but molecular mimicry does not have to be limited to T cells. There is a particularly famous example with antibodies that is shown here. This is a situation where some people will have infection with a particular type of streptococcus bacteria, and they will make antibodies to antigens on that streptococcus bacteria, which you can see here. This isn't all members of the genus Streptococcus, it's a particular specific bacteria. However, it turns out that one of the epitopes that is bound by those antigens actually has there's a very similar protein in the heart. And so the antibodies, which are in the blood, which goes through the heart, will actually start to bind to this antigen in the heart and start to lead to damage to the heart. This is a disease called rheumatic fever. And so in some individuals who are infected with a particular streptococcus species, they will make an antibody response to an epitope that will then lead to heart destruction through rheumatic fever. And now for the rabbit hole. So the other sort of way that we often think about breaking tolerance is related to clonal deviation and clonal suppression. One part of the example that I'm going to tell you about, again, a part of the example that I'm certain that none of you will have any questions about whatsoever, was sort of originally thought of as a deviation Th1 versus Th2 response. I will say, however, that with the example that I'm going to be talking about, I'm going to tell you kind of about sort of our modern understanding of it. It used to be Th1, Th2. Now it's a little more Th1, Th2 Tregness. And so whether we want it to be deviation or suppression, I'm just going to kind of lump those together at this point. And so if you recall, the idea here is that we have different helper T cell subsets that are produced when a different set of cytokines are made by our antigen presenting cell. And so sort of the cytokine environment that the T cell gets stimulated in influences the function of the T cell. The T cell gets activated in one cytokine environment, it might become a Th2 cell. If it becomes activated in another cytokine environment, it might become a Th1 cell. And in different situations, those might be useful or not useful. So one particularly famous story or famous kind of outcome of this that we thought about is shown here. And so this also, all of the rest of the examples I'm going to tell you about today are a place where things get really muddy because whether I'm talking about autoimmunity or allergy kind of gets all mushed together. So yeah, some of what I'm going to say right now is about allergy. For the rest of class, some of what I'm going to say is about allergy, but I just can't. I tried to separate the examples, I couldn't do it. So we're just going to go with it. So this is actually a situation with food antigen. In particular, this example is an example with gluten. And so this is an example with celiac disease. So normally, what should happen when you eat gluten is that gluten should go into your GI tract. And so it's shown here as dietary antigen. It should eventually maybe get picked up by a dendritic cell. And that dendritic cell should turn on a T cell so that T cell can become a Treg. So we should sort of, and in fact, this is usually what happens to most of the things you eat, right? Is that you're going to make Tregs because you don't want to kill bread. You don't want to kill steak. You don't want to kill all the things you eat. And so in general, you probably, the first time you eat stuff, the first time you eat each new thing, some dendritic cell is going to stimulate the T cell that responds to whatever that food is and be like, this is an okay thing, we're going to make Tregs to this. We're not going to destroy the steak. But if you are very unlucky and happen to have a viral infection in your GI tract, at the same time, your body is first encountering that food antigen. Now, here's the dendritic cell interacting with the food antigen. Here's some other dendritic cell interacting with the virus. This dendritic cell is going to make so many cytokines. It's going to be like, guys, we need a massive Th1 response. Now those cytokines might get in the way of this T cell activation. This T cell that should have become a Treg might now become a Th1 cell that responds to gluten. Because you were unlucky enough that the first time you turned on a T cell in your GI tract in response to gluten, you also had a GI virus, this virus called reovirus, that particular day. And so your T cell that should have gotten tolerized in response to gluten, instead that T cell became a Th1 cell because it had a bad day. And you can see that on this slide as well, or this image as well. So normally, here is the gluten. It should interact with the dendritic cell. We should get regulatory T cells. But in this case, that dendritic cell also perhaps saw some virus. That dendritic cell started making the wrong cytokines. And so when it activated the gluten-specific T cell, it pushed that T cell to be a Th1 instead of a Treg. Now when you see gluten again, you don't have Tregs, they're going to keep your response chill. You have Th1 cells that are going to make inflammatory cytokines and lead to this whole process of degradation. And so you can see that sort of the wrong infection at the wrong time, when the wrong T cell, who had not yet been tolerized, was present, could lead to this kind of sort of mistake with or break of anergy. Yep, Brian.
So when you say degradation, like it's not just of the food antigen?
So I'm sort of skipping over some of the A and P parts here, but really what happens is the inflammation actually starts to destroy the GI tract. Because you're seeing so much food antigen, you're getting so much inflammation, you actually start to get destruction of the intestinal wall. Yep, Shelby.
Why is it that people can grow out of food allergies?
So that's definitely something that people are thinking a lot about. There are a bunch of allergy questions that I'm not going to be able to answer, but it does certainly seem like perhaps if you maybe made a Th1 response at some point in your life, over time you may either exhaust those cells or those cells might get pushed more towards Treg. And so there are ideas of both of those. Some allergy treatments, the idea is that we're trying to either exhaust or sort of push towards Treg. And so that's probably the answer, but can't give you a full answer. Yep, Juliana.
So when the dietary peptides enter the GI tract, there's already that large amount of cytokines in there.
Well, it's sort of like if they're happening at the same time. So this dendritic cell is getting the dietary antigen and the virus. And it's making cytokines because of the virus, and so those cytokines are impacting what happens to the T cells to the dietary antigen. Yeah, David.
So again, for this case, it comes down to a naive T cell reaching it, and that hasn't already differentiated into a Treg. And
Yep.
It's kind of like a timing thing.
Yep.
Okay.
Bad luck. Josh.
So if the first time you see food, you make Treg, and then the second time you happen to have the virus and see food again, like with the Treg they make the first time have any influence in food, or would you get?
Sorry, tell me this scenario again.
So you consume food the first time, you make the Treg. The second time you consume it, you have this virus. Would you develop an allergy to it?
No, so you're going to make a Th1 response to the virus, but since you've already started the Treg response to the food antigen, you're going to keep on the Treg response to the food antigen. So it's just if you were unlucky enough for that to be your first time. Because I'm definitely not going to make it through all of the next stuff. I'm going to tell you some more cool stuff, but I'm not going to make it through all the cool stuff. So one of the things you might be aware of, this is going on totally into allergy, but it's related to what we're talking about. You guys might find it interesting. One thing that's going on that's sort of implied in what I'm telling you here is that when you eat antigens, when antigens come in through the GI tract, it seems as though we mostly push things in the GI tract towards Tregs. So usually if you see an antigen the first time in your life through the GI tract, you tend to make Tregs to it. And if you see an antigen for the first time somewhere else in your body, you don't make Tregs to it. You make like Th1s or something to it. That's a big oversimplification. As you might be aware, there have been increasing numbers of people who have peanut allergies, right? one of the big hypotheses about what's happening with peanut allergies is that it used to be that the first time your body ever saw peanut antigen was when you ate some kind of peanutty food as a baby, right? And so the first time your T cells got activated in response to peanut antigen, they became Tregs. Make sense? And so then you don't have peanut allergy because your T cells respond by being Tregs and being suppressive. However, increasing numbers of baby lotions have peanut oil in them. And so many babies are now seeing peanut antigen not through their GI tract for the first time, but in the skin for the first time. And so their T cells are not responding by making Tregs the first time they see peanut antigen. They're making Th1 responses or Th2 responses the first time they see peanut antigen. And so that actually works out pretty perfectly with some of the changes we're seeing in peanut allergy. You can imagine that the recommendations on that are to use lotions that don't have peanut protein, peanut oil on the babies, and also actually to feed babies peanut-containing food really early, to make sure that the first exposure is getting them Tregs before they maybe respond to this lotion. Yep.
Would there not be like a similar issue with other vegetable oils that might be in lotion?
It could be. Then it sort of comes to this question of how easy is it to make an allergy? You know, what is the antigen close to being a mimic? Is it, you know, so it starts to think about, well, what's the allergen, and also like, do we diagnose those and things like that? But potentially, sure. I haven't heard that, but I could see that makes sense. One also related thing is that people realized that in certain areas of the world, and I will say more about this at a later date, the same number of people were positive for COVID, the same number of people were getting infected, they weren't dying as often from COVID. And so in certain parts of the world, specifically helminth endemic regions, people didn't seem to die when they got COVID as much as they did in other places. And there was sort of a hypothesis about this, and this paper is one of the papers that actually really looks at the data nicely and shows that people who were infected with parasitic worms, with helminths, who already had a parasitic worm infection when they got COVID, made a Th2 response to COVID. Whereas people who didn't have a parasitic worm infection made a Th1 response. And the Th1 response caused more damage, and the Th2 response helped you respond better. And so it turned out, if you had a helminth in your lung when you got COVID, your response was deviated to Th2, and you actually didn't have as much damage and didn't have as severe a disease. And so this is sort of another example of that clonal deviation situation. There's another really, really famous sort of story related to clonal deviation / clonal suppression, also related to helminth infection, so I'm going to have to start next time by saying a few things about helminths and helminth infection. But that'll be a next time problem, so be ready for that, and I will see you guys on Wednesday, and remember that your lab report is now due next Tuesday by the end of the day.